Thank you, Joel. Thank you, Julie. And it is a pleasure for me to be here and to share with you the results of my work on protocaterin 19. Yeah, okay, thank you. I'm interested to study the molecular and the functional role of protocaterin in the brain. And just to start and to give you a brief introduction about the synapse and the neurons, I want to explain you that in the neuron we can distinguish two different types of synapse, the excitatory and the inhibitory synapse. And it is important for the normal cognitive functions, and it is important that there is a perfect balance between the excitatory and inhibitory synapse. So, recently, in the last 10 years, many papers demonstrated that in more than 15% of genes mutated in X-linked intellectual disability code for synaptic proteins, and they are involved in many brain many functions, and such as the synaptogenesis or the trafficking, receptor trafficking, and the protein synthesis. So we think that one of them is protocaterin 19, and a couple of years ago we started to study the molecular function of protocaterin 19. So the protocaterin 19, as mentioned, the Tom's before and the speaker before, is located, the genes is located on chromosome X, and the protocaterin 19 belongs to the protocaterin family and is a transmembrane protein. And here you see the structure of the protein. You can see an C-tail, intracellular C-tail, and transmembrane region, and the extracellular loop, where most of the mutations are identified. So the mutation on protocaterin 19 causes the dyslexia and intellectual disability and autism and behavioral disturbance. So our approach, we are studying the protocaterin 19 by using a different approach. We are using the culture neurons to identify the molecular mechanism of these proteins, but we also we are interested to study the physiology and the functional role in the brain of protocaterin 19, and to do that we are using two different animal models, the in utero electroporation system and also the conditional knockout mouse. But we are interested to validate the in vitro study and in vivo study with the in IPS model. So the protein, we don't know too much about the molecular function of the protein, but what we found is the protein is highly expressed in the brain, and in particular in cortex and hippocampus. And as you can observe here, this is homogeneity from rat brain. But if you look at the temporal expression in rat brain of protocaterin 19, you can see that the maximum expression of the protein was observed during the early stage of development. And this was observed in rat hippocampus and also in cortex. And in parallel here, you can see that also in the primary culture neurons, the maximum expression of protocaterin 19 was found during the synaptogenesis. So these results indicate that protocaterin 19 probably is involved in synaptogenesis. So in the next experiment, we analyze the localization of protocadrin 19 in calcium neurons. And first in heterologous cells, and if you have expressed protocadrin 19 in HK cells, you will find that the protocadrin localized exactly in patch in cell-cell junction, and in neurons co-localize very well with markers of excitatory and inhibitory synapse. So you see here in young neurons the protocadrin 19 localized with PSD95 and markers of excitatory synapse. And in mature neurons, protocadrin 19 co-localized very well. You can see here the yellow puncture with PSD95, but also with markers of inhibitory synapse. So, and in biochemical fractionation, subcellular fractionation experiment, we found protocadrin 19 co-migrate with PSD95. So protocadrin 19 is expressed in inhibitory and excitatory synapse. So in the next experiment, we analyze the downregulation of protocadrin 19 in calcium neurons and also the overexpression. We modulate the expression of protocadrin 19 in calcium neurons, and we found that the expression levels of protocadrin 19 regulates the markers in the development of hippocampal calcium neurons. So, if you look at the analysis of Western blots and neurons lacking in protocadrin 19, you can see that there are a downregulation of PSD95 AMPA receptor subunits and also GABA A receptors. So, but if you overexpress the protocadrin 19, you will see also an increase in the level of PSD95 and GABA receptors. So, seems to be the protocadrin 19 seems to be important for maturation and of the synapses. So, interestingly, we found that the C-tail of protocadrin 19 interacts with the GABA A receptor, with the alpha-1 subunit of GABA A receptors. We identified this possible interaction by using a classic screening, biochemical method, the 2-hybrid screening. And we validated this interaction by using two different cell models, the HK cells and neurons. And here you can see that the protocadrin, the overexpressed protocadrin 19, co-localize very well with an overexpressed alpha-1 subunit of GABA A receptors, and also co-immunoprecipitate very well. So, of course, the mutant form of protocadrin 19, this is a molecule deleted in the C-tail was not able to co-immunoprecipitate the alpha-1 subunits. And also here, this is another control that we used, that the PC9 was also unable to co-immunoprecipitate the alpha-1 subunit of GABA A receptor. So, we also validate in calcium neurons this interaction, and the endogenous protocadrin 19 co-localize very well with the alpha-1 subunit of GABA A receptors, co-immunoprecipitate also in hippocampal neurons, and also in homogeneity of mice and and also this is another control that we used, that the GST, in GST pull down, the C-tail of protocadherin 19 pull down the alpha-1 subunit of GABA A receptor. So we are now confident that protocadherin 19 interacts with GABA A, with the alpha-1 subunit of GABA A receptor. But the question was now is, does the protocadherin 19 regulate the trafficking of GABA A receptors? And interestingly, we found that the silencing of protocadherin 19 in calcium neurons causes a reduction level in the surface of expression of GABA A receptors. Of course, the overexpression of protocadherin 19 recovered the normal expression of alpha-1 subunits in neurons. So, and interesting also, the silencing of protocadherin 19 affects the GABA A transmission, because we found that in neuron the silencing of protocadherin 19 they are an impairment in the frequency of in the postsynaptic inhibitory currents. And as expected, we also observed an increase in the decay and area. So this is an indicator that the conclusion of these results are following. In absence of protocadherin 19, there are less synaptic contacts, but they are still work well because we didn't observe a defect in amplitude. But the reduction in the expression level of alpha-A subunits causes an increase in area and decay level. So, but what happened in vivo? As mentioned, we are interested to study the function of the protocadherin 19 in vivo, and we are using two different approaches. So in that case, our collaborator, Laura Concetta in Genova, she performed this nice experiment. She used electroporated in uter and used this technique to silencing with the specific shRNA the protocadherin 19. And then she looked at the morphology and the migration in vivo. And what she found are summarized here. So she observed in cortex and hippocampus a defect in the arborization of the neurons. And also she observed a defect in migration. But all interesting, she also found that the animals are more susceptible to seizure. So the animal, they don't show a spontaneous seizure. But if you simulate with PTZ, you will increase the seizure susceptibility. Of course, these experiments are not complete because now she are doing the rescue experiment, and we hope to recover the normal morphology and migration, and also the phenotype. So, but also we are generating conditional knockout mice. At the moment, we don't have preliminary results about the phenotype, but what we want to do are summarize. The first, we want to characterize the behavior of protocadherin 19 mice. And of course, we want to also characterize the physiological role of the protein in the conditional knockout mice. And in collaboration with Jim Miratto in Pisa, we want also generate an in vivo mosaic in vivo model to study the mosaicism. And of course, we also want to validate the GABAergic hypothesis in that model. So we are unfortunately yet we don't have the results about the iPS cells, but of course our idea is to also have the to develop and to have an human iPS cells to validate our hypothesis in vivo. So, in conclusions, we demonstrated in culture neurons the protocadherin 19 is highly expressed, is localized at the synapse, and is important for the normal synaptogenesis in vitro. And we identify an interaction with the GABA-A receptors, and probably the protocadherin 19 regulates the trafficking of GABA-A in the surface, and also is important for the normal GABAergic transmissions. And in vivo, we also find that we confirm the data that we before observed in vitro, and in particular we found that the protocadherin 19 is highly expressed in hippocampus and cortex, and the downregulation causes and defects in migration and in maturation of the neurons, and also the animals seems to be more susceptible to the seizure. So, at the end, I want to say thank you to the protocadherin 19 aliens that support our research, and also the Acute Syndrome Foundation and the Insieme per la Ricerca PCDH19 PCDH19 onlus for this grant support. And thank you.
Questions?
Nice talk, Maria. Can you go into what the migration phenotype was with the in utero electroporation? What did you see?
You go to the probably they are here. Laura, she observed defects in the migration in cortex and hippocampus. Of course, here this experiment is not complete because we need to rescue the phenotype. But probably there are also defects in the neurogenesis. We don't know, but probably.
Has she tried to electroporate the interneuron progenitors?
No, she didn't
harder
try.
to do,
Yeah. This is a question that she didn't try.
Well, you can't electroporate the progenitor interneurons because they are an area you cannot really reach with the electric field.
You can change the polarity and get some of them.
Yeah,
unfortunately, the parvalbumin are later born, and that's harder to do.
Simple question, Maria. Nice work. Do you see there's a scope for testing some of the PCDH19 variants in your system, for example, with the surface rescue of the GABA receptor alpha subunit expression? Because it'll be valuable to see. I mean, you managed to rescue it with wild type overexpression, but
like you mean to try the other mutants?
just a few
yes,
missense
no,
mutations to see what's going to happen in that case. Perhaps to see whether you can rescue it with a different predicate here in like 17 or 10 we heard.
No, but we are making the construct to do that. Yeah, we didn't yet know.
And I just had one quick question. When you looked at the hippocampi, were the changes symmetric? Just getting to Paul, there's
No,
heterogeneity
actually.
in terms of the population of cells that may express protocadherin.
No difference.
No difference.
No.
And maybe that's why I can ask Paul a question. I mean, did you see any heterogeneity where there was a specific lobe that was very asymmetric? And I asked this just because we have one adult patient in our practice that actually underwent a temporal lobectomy with protocadherin and has been seizure-free for 5 years. I won't go into the details of how she came to surgery, but
we see a wide variety of different patterns of inactivation driving this segregation phenotype. Occasionally we've seen mice that almost have a complete absence of protocadherin, for example, across the whole cortex. But I think that's just due to the random nature of the inactivation itself. So we see a whole variety is all I can really say.
and just while you have the mic, we had a late question come in from the web viewer for you. I'll try to. So, does the HA/flag tag affect the function of PCDH19?
It's a good question, and no, we don't think so.
And then there's just a three-part question. So, have you looked at the red-green brains later on?
Only as late as P7, maybe some adult P42, a couple of samples. Actually, we find that the expression dips a lot by P42. There's still expression in layer 5A, but it's reasonably weak. And the segregation phenotype's present throughout into adulthood.
So that was actually the follow-up question. Do the neurons intermingle postnatally?
Yeah, that was kind of one of the questions we had earlier.
Yeah.
That's probably the key thing to do next, is to work out what the connectivity changes are that are associated with this early segregation phenotype.
Great. Any other one question? Did you have a question before for Dr. Thomas? Just